Okay, so I would like to present the last speaker, Pinar Ozbay from the Bogazi University in Istanbul, and she will talk about contributions of the autonomic nervous system to brain dynamics during sleep and wake.
Is it this thing that I should use? No, that doesn't look like a pointer, is it? Okay, that looks, okay. That looks a bit scary. Let me. Okay, that'll work. Well, I'll talk about sleep. Where am I going? No. Can I go back now? Oh, okay. Thank you. So, okay. Thanks for staying awake and considering that we had a coffee break without coffee, indeed. And so today I'm gonna talk about autonomic contributions to brain dynamics during sleep and wake. So I'll go through fMRI, human nervous system, physiology, CSF, EEG, and many more actually examples from the literature. So we had a great lineup, I have to say. Initially, I was worried I'm giving the last talk, but now I see the reason because I have really nice already like introduction from our colleagues. And so the fMRI, as we all know, is like a response to a brief local neurovascular dilatory event. And we have blood volume changes, blood flow effects, and it's all a mixture of combined both task and physiologically related signals, actually. And we also discuss noise throughout the day here. And we have various factors contributing, like thermal noise, system noise, and maybe artifacts, we can call them, due to head motion, respiration, heart rate, other physiological processes. And we are trying to understand, and after we collect our data, kind of a backwards engineering, actually. So just a bit overview on the human nervous system. So we have the central nervous system and the peripheral nervous system. And the first one comprises mostly brain and spinal cord, as we all know. And the peripheral nervous system that I will mostly discuss today, and its effects on the fMRI and brain dynamics. So it has another two branches, somatic nervous system, autonomic nervous system. And the latter is again the where that we are, or I am more interested in. And two more branches, sympathetic nervous system and the parasympathetic nervous system. So the somatic nervous system, just briefly, is responsible for carrying sensory information from body to CNS, basically. And it's extremely important because it is kind of help us to detect like the touch, pain, temperature, and many other motor neurons. And the autonomic nervous system, we had also a nice introduction about that. So it has two branches, sympathetic and parasympathetic. As you can see, those are all the vital functions that we need for our daily life, basically. One part is sympathetic system is the, I'm so worried, is it the green, the pointer? No, the pointer, the colored thing, the dot. Well, we are doing high-tech stuff, but here I'm struggling. Okay, here the tiny, anyway, I give up. So we have the sympathetic system. Basically, once it's triggered, it's like fight or flight, and pupils dilate, and heart rate increases, and it causes release of adrenaline, and like related to stress hormones and so on, and also blood pressure increases. And on the other hand, there's the parasympathetic system, but those are not like on and off together. They actually kind of go together throughout the functioning. So on the parasympathetic side, it is like slowing heartbeat and relaxing, pupil constriction, and so on. And we are going to discuss a bit more examples covering those systems. So the second one is also called like the rest and digest. And basically the terminology tells what its functions indeed. It is slowing the heart rate, stimulates digestion, and promotes relaxation, basically. But I mean, you can think like in fMRI or MRI conditions, it's mostly what we are discussing is the sympathetic one, because I mean, it's hard to, I think, generally relax in the MRI overall. And once we discuss central and peripheral nervous system, they, as we said, they work together. They coordinate also physiological processes throughout the body. And so overall, today we are going to try to understand, dive a bit more the autonomic physiology and its effects on the brain signals, basically. So we have emotions, and they are still ongoing while we are in the scanner as well. We also can have some tasks going in the scanner, and they can create fear, and anger, anxiety, and they can result in increase in heart rate, rapid breathing, and or release endorphins. It can be a memory task, or decrease in heart rate, or we may get into a deep sleep, although it's a resting state, open eyes, data, for example, we cannot stand, and we may have to a deep sleepy mood, and then we shallow our breathing and so on. Those then all actually affect our signal, and we have to be aware of our physiology to better understand the fMRI signals indeed. So let's say we have a subject in the scanner, we have a 3 Tesla, so we ask subjects are to stay awake or sleep, or stay focused, or relax, or alert, stress, or lots of tasks that we can ask for. And of course, those are all like affecting our respiration, cardiac regulation, and also blood pressure. And they then like always keep interacting each other throughout the scans. And things get even more complicated in sleep, and especially if you want to study sleep in fMRI. So, because there are like lots of sensory and physiological stimuli can affect, and in this review paper, it's nice to discuss how many pathways can be involved actually in the regulation of systemic physiology, and which regions are being also affected due to that, like including cortex, thalamus, amygdala, hippocampus, from like subcortical and also from brainstem, and pons, medulla are also regulated through sympathetic activity, respiratory activity, motor activity, and so on. So a little bit, I'll give introduction to sleep, because I thought not everyone knows about sleep stages and what they actually affect, how they affect our physiology. So as we probably all know, circadian rhythm refers to the 24-hour internal biological clock, and it affects our daily life. I'm sure half of us are under jet lag now, so it's really critical for our daily being. And basically, our physiological processes are being affected highly by our sleep cycle. So let's see like a really brief example how the stages differ in the EEG data. So EEG, like a really trade kind of the marker for sleep scoring, basically. And from top to down, we can start with the wakefulness, the relaxed state, and stage 1, we see the theta waves, and stage 2, as we see markers like sleep spindles, K-complexes, we are gonna learn a bit more about those later, and the last one is the slow wave sleep, the deep sleep. So as the names actually coming from, the signals actually reflect their names, so the signals are also becoming slower and larger in amplitudes. So just as an introduction for the stage 1 sleep, we call it part of the light sleep, and it's the first stage of non-REM sleep of the cycle, and we are beginning to relax, and the waves are getting slower, and then decreased muscle tone is observed, and decreased reaction to stimuli. Things getting more relaxed in the stage 2 sleep, and body temperature drops, heart rate drops, and this is a longer and then the stage 1 sleep, definitely. Nowadays, many of us have the watches as well. They can also even track our sleep. You may see light sleep, for example, including stage 1, stage 2, goes up to more than half of our sleep, indeed, during the night. And it's a deep state of relaxation, and of course, becoming more difficult to wake up. And there are some markers of light sleep. One is sleep spindle. You can see the distinct pattern of that. And the other one is the K-complex. Both are really critical for memory consolidation. So this latter one, the K-complex, is a large wave, you can see, and if going shorter than a spindle, and ranging in frequency like 0.5 to 2 hertz. So those are also being linked to various cognitive processes and memory consolidation. And lastly, no, not lastly, but for non-REM sleep, last one, the deep sleep. So this is like even further brainwave slowdown and then body experiences a state of deep relaxation, and the waves also becoming really slower and larger in the amplitude. So the REM sleep, we are not going to touch a lot today, but still I will show a part of it from our data. So the REM sleep, as you can see, stage of sleep, as we all know from vivid dreams, and it's the other main part of the sleep cycle, one being non-REM, the other REM. And I also wanted to share a book that I really enjoyed reading by Matthew Walker, Why We Sleep. So if you're interested in sleep and neuroscience, it's a really nice book to read. So in coming to REM sleep, we then observe increased heart rate, blood pressure, breathing rate, and looking at the EEG data, actually it really resembles the awake indeed. There are also markers of REM sleep, and those are like sawtooth waves. Of course, from the name, we can understand rapid eye movements, so we can also see the eye movements from our EEG channels, and also what is important is the absence of muscle artifact. Okay, so let's come back to our fMRI physiology and sleep relation. And so during our scans, today we learned a lot about physiology nicely, so we can collect various signals during our scans. So those could be respiratory bellow, pulse oximeter, and also not largely discussed today, blood pressure. I had some experience collecting blood pressure data during fMRI, so please approach me if you were successful collecting good quality of data, to be honest. I was, to be honest, not. Okay. And it was really tricky. I mean, the hardware seems practical, but when it comes to the collection data, it was tricky. Anyways, but still I will show an example today. So those are the early papers and well-known papers from Byrne, Bright, and Zheng et al. They are discussing the RVT relations during normal breathing from the left, and then deep breathing. We see that white matter's gray matter pattern, positive, negative correlation in the whole map. And then on the left here, you see on your right, the RV heart rate percent signal variance explained in the rest. And those are from 3 different subjects. You can see that the signals are highly also related to, mostly in the gray matter, positively in relation to respiratory volume and the heart rate. And this was the blood pressure example that I wanted to show you again during rest. And those are group mean arterial pressure and fMRI correlation maps. So what we see here, generally the similar pattern, right? That the pattern of CSF ventricular white matter region with the gray matter. And that there is also a lag between those signals. One is the preceding, but depending on the location of the brain, actually, that lag also differs a little bit. So what else we can learn about that physiological signals, actually, I will discuss today, is also another measure from the PPG. So traditionally, heart rate variation, or the heart rate we do calculate for our fMRI, data analysis and physiological interpretation. But what else we have is the changes in the amplitude of the PPG signal related to the finger skin vascular tone. So like a drop, meaning a vasoconstriction, and at the largening, meaning peripheral vasodilation. And we discussed partially those. So those are again like a marker of indeed sympathetic and parasympathetic activity changes. So not a lot of studies have actually discussed so far that finger skin vascular tone, and those are the 2 examples, again showing large variations across brain. On one side from Hood et al. 2016, and it's quite varying across subjects, which was not fully explained indeed. Those are like awake subjects, actually, and pulse height variability, they called the PPG amplitude. But as you can see, the correlation with the fMRI was really different across subjects. On the other side, again, a peripheral measurement, they calculated, and those are ICA, so the independent components. And similar to the correlation maps, they observed one sensory-related positive correlations, and also, like, again, a white matter ventricular region popped out as a negative component map. So coming to the sleep study, so I will hopefully merge those together. So here is a study that we conducted at the NIH. The study is still ongoing. This is a simultaneous study, EEG, fMRI, and overnight study. Subjects, believe it or not, stayed in the scanner up to 8 hours, but they were allowed to, of course, leave and give breaks. And then that allowed us to actually capture a whole sleep cycle throughout the night, from wake, stage 1, stage 2, deep, and also REM. So this is just one EEG spectrum that I wanted to show you. And here in this data, so this was supposed to be a video, let's see. Yes, it's a simple video showing the PPG amplitude and the fMRI correlation, basically, during a light stage, light sleep stage. So that pattern was there again, and that brought us the question that what is the reason behind that. And actually those were always coming up with the increase in the heart rate, of course, related again to sympathetic and parasympathetic variation. And also in the literature, it was shown that EEG arousals indeed were accompanying those PPG amplitude drops. So those EEG variations together with the PPG, and then once we checked our EEG data as well, we also observed those. They were actually quite K complexes. And those were the data time points always mostly co-occurring during the light sleep, stage 2 sleep. So what is a K complex? It's the signature of subcortical arousal, marker of stage 2 sleep. So we wanted to deep dive a bit more to the data and see how it's corresponding with the physiology, EEG, and fMRI, and try to understand how much it's coming from the neurological side and the physiological side, actually. And in the literature, it's been discussed a lot, the K complex from the EEG perspective, like a marker like for subcortical arousal. And it also has like physiological mechanisms really affecting various parts of the physiology, as again discussed in this review paper, sympathetic and cardiac vagal outflow is one part, and also respiratory gating, barrel reflex. There are like lots of branches that one has to really discuss once covering such a topic. So in this paper review, they also covered that. So during sleep, they sent an acoustic tone. Basically, they triggered a K complex. So this is not like naturally happened K complex, but K complex elicited by the tone. But that was actually accompanied with a heart rate increase again. And so that is kind of the similar background with the PPG drop. Because actually in the heart rate, an increase is initially due to sympathetic increase, and then the drop is due to the parasympathetic variation. So those are like both reflected in the heart rate signal. So coming to our data, we did analyze our sleep data and calculated low-frequency EEG power as a marker for slow wave, so the K complex, and PPG amplitude and fMRI in the whole grain matter. So you can check the signals, just an exemplary from one subject. Then we basically calculated correlations like a legged correlation manner. So we kept one signal same and shifted the other and tried to understand their relations in order. So one example, like this is what it popped up, like on top, low frequency EEG, PPG amplitude, and fMRI. Think it like a matrix variation. So for the PPG low frequency, the 0 point you can think like is the moment of the K complex, and then we have a negative correlation, meaning that a low frequency slow wave increased power is then preceding a PPG amplitude drop and then fMRI gray matter drop, basically. Hope that was informative. And of course, if we cross-correlate PPG and fMRI, then we have a positive correlation. So the legs are critical here. We are gonna come to those later. And if we do that calculation in a voxel-wise, so basically calculating cross-correlations at each voxel, then we had that distinct pattern again between both EEG fMRI and PPGM fMRI, so pointing to a common origin, actually. So now, the autonomic activity that I will try to briefly mention and the CSF pulsations during sleep and wake. So the CSF has been nicely covered with Laura, so I will skip forward here, and then that beautiful paper coming from her group actually really paved the way for the CSF research. And then in this work, they showed like really consistent patterns, although that positive and negative variation in the fMRI in gray matter versus the CSF. And again, that also gave another rise to clinical cases. For example, in this one, the Alzheimer's disease, they discussed this pulsatile CSF movement may be assisting actually glymphatic clearance, and they observed reduced coupling with the Alzheimer's disease-related pathology. Then we checked our data. Okay, we asked ourselves, okay, what's actually happening in the CSF in our data? Because we had like 12 healthy subjects and 60 hours of data we scanned throughout the night. And this is one example from one subject: EEG spectrum, slow wave activity, chest belt signal, respiratory flow, PPG amplitude, CSF signal, and global signal. So you can clearly see probably the light sleep, how the signals vary largely together. And in deep sleep, for example, that is basically diminishing. So there is something going on, definitely in the light sleep part, which we discussed before as depending on the cortical arousal. And on the right is a part that is focused, showing like how they co-vary together, the K complex, increased respiration. The vasoconstriction is nicely clear in the PPG raw signal as well. Then we analyzed those 60 hours of data from our 12 healthy subjects. And this came out basically. Correlation of fMRI with the slow wave activity across sleep stages, top to bottom, deep sleep, light sleep, wake, and also REM. So what is interesting here, we calculated those correlation maps with legs. So one part we called neurovascular leg, the other is like the autonomic leg. And basically we observed a relatively strong negative correlation, for example, in deep sleep, and also strong and more widespread correlation at a leg of later, like 12 sleep. So definitely not the hemodynamic leg, for example. Mostly relevant for stage 2 and stage 1 sleep. So we thought that this can be more related to an autonomic contribution. So since we thought it can be related to a physiological autonomic contribution, then we decided to make like an autonomic correction and wanted to see how that affects the outcome actually. So this basically came out following our physiological regression. Like on the red plus, we see the correlation SWA with the CSF, and also other one with the global signal. So basically, significant correlation we observed, except REM for the all arousal states. And once we remove the fMRI for autonomic signals, so it is nicely discussed the modeling part, for example, for the physiological regression. So we included the respiratory and the PPG amplitude signals for the autonomic correction, and we observed significant reductions in the correlations. But what I wanted to show you here is the difference between 9 seconds and 12 seconds. But that actually shows us a shorter leg for deep sleep may signify a joint contribution for the neuronal and the autonomic pathways. So if it was purely neuronal, we would expect probably more like a 5-6 second delay, but it is not fully physio-autonomic contribution as well. So there's probably a joint contribution of both sides. So, yes, basically we should be collecting the most data we can to understand the signal we are basically analyzing. Well, more papers came out, so I will briefly go through a couple of papers came from like autonomic perspective and sleep. One is this one, human non-REM sleep. They check brain-wide motor and respiratory pulsations. Here in this work, they also used a fast fMRI sequence called MREC. And actually they showed, probably it looks pretty small, but basically from awake to sleep, they again observed like really large pulsations and variations in the signals, and both time and frequency domain data, they showed increased power and the amplitude of brain pulsations basically, and they verified again the sleep stages with the EEG that was also predominantly a non-REM stage 2 sleep. And basically what they discussed here, the global very low and respiratory frequency power, they observed increase markedly in sleep versus the wake session. And they continued further, before continuing, sorry, one last thing that I want to say, basically here they discuss physiological pulsations may overpower the neurovascular coupling, and actually that may be affecting the enhanced brain clearance. And because the brain regions that are mostly was affected were around actually sensory regions, indeed, in the maps. So if you're more interested, there is also a sleep deprivation paper actually that even like made the results even more like stronger, and that was also really interesting. And that was the resting state paper, another one from discussing PPG and CSF signal variations. So it doesn't have to be sleep. So here I will show a really brief examples during wakefulness. And they also observed high correlations between PPG-related signals and the fMRI. But also related to what Laura recently discussed, they observed variations in the CSF region. So if you want to do CSF-related regression, we have to be careful because actually the signal is not really constant across different parts of the CSF, like the ventricles. Another example was using fMRI signal like a pattern, like a template, to regress out from the resting state fMRI. And here, regressing out that information such as physiological regression also reduced greatly the correlations between EEG and fMRI. So this was again a resting state study where mostly focusing on the arousal changes. So here they showed heart rate, RVT and EEG and fMRI cross correlations, and all of them, the top ones are before and later ones are the after regression, and you can see clearly the correlations are diminished. So I think this should be the last one. Okay, I have like 3 more minutes, I guess. For autonomic correction during alertness, so this figure is actually from the supplementary part. What they showed is actually maybe related to the cognitive question that one of the audiences asked. So basically, once they removed the RV and the heart rate in the regression in the model, the alertness index relation between fMRI EEG has actually reduced. So such things actually are respiration and heart rate related signals probably are actually informative about our behavior and analysis for like prediction, for example, on the alertness measures. So last but not least, lots of other tasks we can design to stimulate sympathetic activity. So those can be pupil dilations that we also showed recently. Following deep breaths basically. So following deep breaths, we observed also PPG amplitude drops because deep breathing is also an affecting factor for the sympathetic activity, and also an increase in the CSF signal we observed. So this was more like a purely physiological effect on the CSF signal. And also a continuing study, like a cognitive task, I presented last year here at OHBM. So this is like a cognitive task based on a summation, so creating stress in the scanner. So here what I tried was isolating the RVT-related effects, basically. So I basically created events with and without large respiratory variations. What happens here is, regardless of a large RVT changes, we still have a pupil diameter and fMRI correlation, actually. Still have the amplitude drop, pupil can dilate, regardless of carbon dioxide-related variations. So what this tells us even further, after I checked the CSF as well, even we had the increase in the ventricular CSF region. So actually pupil and PPG amplitude can explain additional variance, and those can be also combined for our models. And indeed, PPG amp may be complementary for our pupil studies as well. So take home, impact of autonomic physiology is definitely significant. And we have to account for those, and we have lots to measure. And if you want to hear even more, so we have a couple of abstracts discussing caffeine as a sympathetic stimulant, and also like autonomic correction, including PPG and pupil diameter. So, and hopefully a fun week is waiting for us. Thanks for the organizers and everyone, my previous lab, Jeff, and of course the whole lab there, and my current students, and you for your attention.
Thank you very much, Pinar. We have a question online for you. What do you make of a recent Nature Neuroscience paper by Miao and colleagues showing that brain clearance is reduced rather than increased during sleep with fluorescent dye in mice?
It's reduced?
Yeah, they say that this study shows that brain clearance is reduced rather than increased during sleep due to, and this study has been done with fluorescent dye in mice.
Oh, okay. I should recheck that paper, but what was the sleep stage there? Do they maybe? we can discuss, or were they naturally sleeping? Probably, right? Not anesthetized. I have to reread the paper. I remember that one coming up, but to be honest, to answer better. Yeah.
Okay. In that case, I think we can all thank you again for your great talk.